Okay. We have almost made it through to Thanksgiving. Just got today and Monday left. So we are going to continue talking about muscles. We'll see how far we get today, but we may have some interesting new things related to muscles to look at on your turkeys, if you're eating turkey. Any questions before we get rolling? Okay, cool. All right. So when we left off, we were finishing up our tiny last little bit of our description of whole skeletal muscles and how they're used before we dive into some details of their physiology, particularly how they are getting energy. So we're going to talk for most of today about skeletal muscle metabolism and how that works. Okay. I want to make sure that we finish up what we were doing last time because it's going to come back a tiny bit after we tack on this new metabolism information. So we talked about the fact that we can change how strong muscles are, how much force, how much tension they generate in a couple different ways. So we can recruit larger motor units. We can recruit motor units that have fibers of larger diameter in order to increase strength. And we had just started to talk about that piece about motor units of different size. And think about the fact that with any given muscle, you're going to have multiple motor units in it. So motor unit is a motor neuron and all the fibers it's attached to. So we can recruit them in order of size. We're going to start with the little ones, and then we're going to build our way up to larger motor units. And this is both useful in the day-to-day, so you can kind of be as lazy as possible when you're trying to move something. Use more controlled, smaller forces if something's lighter, bigger, pulsive forces if something's heavier. It's also related a little bit to their structure, because when we think about how those sarcomeres contract, in order to contract more sarcomeres in a larger muscle, we'd ultimately need to release more calcium. So that's also part of why it takes a bit longer to get larger motor units on board. Okay. So our order of recruitment of motor units is based on their size. Small ones first, larger ones stepping up in order of size. So our larger neurons, one thing about them, right? A larger neuron, when we're thinking about a motor neuron, right? You're going to need a larger neuron if you're going to split out to more muscle fibers. Okay. So if you split out to more muscle fibers, that'll be a larger motor neuron, a larger motor unit. It's going to take a bit more time to depolarize that to threshold because we have to spread around those charges through a larger kind of space. So this is going to require more synaptic input, so more input from the neurotransmitters earlier in the chain controlling that neuron, versus our smaller neurons are going to be excited more rapidly because they're smaller, there's less space that you have to spread that depolarization across, so we can get them on board a little faster. So if we're looking at recruitment, we talk about something called the size principle. All the size principle is that we're recruiting those motor units, small first, biggest last. Okay. So here we can see our first smallest motor unit, right? Over time, it's going to start us off building tension in the muscle with this little motor neuron X, and therefore this little 3-fiber motor unit. So because it's small, we got a little bit of tension. Now we are generating more and more neurotransmitters, synaptic input here. We can eventually get this middle motor unit on board. So we can see that this has slightly larger fibers, has slightly more fibers, produces slightly more tension. And we're not turning off the smaller unit before we do that middle one. We're stacking these on top of each other because we're trying to build up to enough force to do whatever it is we're trying to do. Yep. So the larger one comes last. Okay. So now we're going to switch to talking about metabolism. After we talk about metabolism, we will link this a little bit to those side principles. Flip into our next packet. So since we're talking about metabolism, we're going to do a brief review or brief touching back on some of the parts of that first unit that are going to be relevant to muscles specifically. So go ahead and join me here, and we'll do a couple of metabolism. If we don't remember metabolism, it's going to be harder to follow what I'm about to say. All right, let's get started. The first thing we want to remember, what molecule was it that we were oxidizing during our aerobic cellular respiration, our typical normal metabolism, in order to get energy for ourselves? Okay, yeah. So the thing getting oxidized is the glucose. So sometimes we refer to our typical form of generating energy as glucose oxidation or glucose catabolism, right? So this is why we needed our reduced electron carriers from earlier steps to go into the mitochondria and fuel our electron transport chain, because this whole big picture is all about oxidizing the glucose, getting out the energy in the bonds of glucose. That's why we also spent time thinking about when do we have glucose in the bloodstream, how much, because glucose is really critical for energy production in the form of cellular energy, ATP. So ATP was what we were making. Reminder, where do we have glucose stored? What's the name of our big polysaccharide for glucose storage in us, because we are It'd be different if we were. Okay. Yeah, so glycogen is our storage molecule in the cell for glucose units. So glucose can be strung together, those glucose monomers, into a polysaccharide. Glycogen, glucagon was one of the ways that we were releasing glucose by breaking the bonds in glycogen. So glucagon was our hormone associated with the release of the glucose. Glycogen is our storage molecule. In a plant, it would be starch. So it's like your version of starch. Okay. Thinking about the steps of metabolism, cellular respiration, which part of it was it that made us the most ATP? Yeah. So all three steps that are on the board right now to generate a little bit of ATP, but it's oxidative phosphorylation using that electron transport chain that really gives us the bang for our buck. So the glycolysis, we got 2 ATP on net. The Krebs cycle, or the TCA cycle, the citric acid cycle, happening in our mitochondria mainly is giving us a lot of reduced electron carriers. We're also getting a tiny bit of ATP, like one for each circle of the Krebs cycle. Oxidative phosphorylation is where we have this big explosion as we release our proton gradient, our hydrogen ion gradient from that intermembrane space, letting it flood through that inner membrane, spinning around our ATP synthase, making sense of ATP. Cool. So which of our energy generation processes can we do without? Presence and absence of oxygen is going to be important for us. Yeah, so the answer is glycolysis. So glycolysis is the first step of breaking down glucose. Glycolysis is the one that's happening out in our cytosol, in our cytoplasm. So all we need are enzymes in order to do glycolysis. We don't need mitochondria. It's not happening inside a mitochondrium. We don't need oxygen. We actually don't use oxygen until all the way at the end. Our oxygen is our final electron acceptor at the end of the electron transport chain. So we need oxygen directly to do oxidative phosphorylation. You need it kind of indirectly for the Krebs cycle, not involved in the cycle itself. But if we don't have oxygen, we don't do oxidative phosphorylation, and therefore everything gets backed up inside the mitochondria. So ultimately our Krebs cycle wouldn't be working either if we didn't have oxygen. Beta oxidation of fatty acids is a different way of getting energy, unlike a triglyceride. And it would be using some of these same processes. It also leads us into our oxidative phosphorylation eventually. It's glycolysis that we can do separately from the other steps without oxygen. Okay, cool. So congratulations. We got a bunch of people who got those right. If not, hopefully we've jogged our memory on how this stuff works. Because what we're going to be doing next is thinking about our skeletal muscle and thinking about fiber type. And so we have motor units attached to our motor neurons, but we're also going to have some differences in the muscle fibers. And those differences are going to be related a little bit to how strong they are, how fast they react, and how they get ATP and how much. Right. So here's our general review up over on the side here, right? So what we're doing when we're making ATP is we're taking glucose, which we may have gotten from a storage molecule called glycogen, might have gotten it directly from our bloodstream. And in the cytoplasm, we can do glycolysis, giving us a little ATP to on neck. We then need to convert glycolysis into pyruvate. That's what happens when you split like our glucose molecule in half, right? Ultimately, this is a slightly more stable form, okay? And if we're going to continue into our whole aerobic cellular respiration process, we're going to pull that pyruvate into the mitochondria, peel off some carbon dioxide, make our acetyl-CoA, and that's what goes into our next main step for this class, that Krebs cycle or citric acid cycle, producing a tiny bit more ATP, but we're inside the mitochondria now. And then oxidative phosphorylation at the end, using oxygen, giving us lots and lots of ATP. You can also see how the fatty acids would fit in on here, right? So those fatty acids, if we're doing beta oxidation, don't do glycolysis. They jump in to those processes occurring inside the mitochondria. So a little bit into this Krebs cycle, a little bit here right before oxidative phosphorylation. So we care about this because in order for a muscle to contract, we need that energy to do the work, right? And we saw where that ATP is used when we were looking at the cross-braided cycle. So we saw up at the top of that cross-bridge cycle, we were using that ATP, breaking it down, so doing a hydrolysis reaction, breaking the bond between our final two phosphate groups, leaving us with ADP and that phosphate ion. That was what led us into that sort of copying of the cross-bridge that headed the myosin molecule into this high energy form, which is the form that we need if we want to start pulling actin towards the middle of a sarcomere. So we need ATP. Which means we need to make ATP. We need to get it from somewhere. So in our muscle cells, we are going to be making ATP. So we're not getting ATP from the bloodstream, it's happening in the muscle fiber itself. So ATP is driving that cross-bridge cycle. So we need to make ATP inside our muscle fiber. We've got a couple of ways that we're going to be able to do that in any given muscle fiber. Okay. So actually going to read this from the bottom up, right? So our first way over here, when we were thinking about metabolism, was that glycolysis, which is anaerobic, no oxygen needed, right? The second way we talked about when we talked about it before was oxidative phosphorylation. So we do need oxygen. We're inside the mitochondria, right? Later on in the process, but more efficient, more energy. And the reason I'm reading them backwards is we haven't talked before about this first one. Okay, so the phosphorylation of ADP by creatine phosphate. Okay, so when we say phosphorylation, all that means is tacking on a phosphate, right? So we know that ADP is adenosine diphosphate, right? So two phosphate groups. So if we phosphorylate it, we are adding on an additional phosphate, and now we have ATP. Okay. So this is not a process we've talked about before, but it is something that can happen in your muscle fibers. It does happen. This is actually kind of an immediate form of on-tap energy production that we have without running any processes. So it's limited, but we've got it. So when you talk about taking like creatine supplements, this is really the process you're kind of trying to juice up. Okay. So here's how the system works. Actually, let's take a look just at the bottom. So we have some creatine phosphate present in a muscle cell. So you can see that down at the end. Okay. What happened here? We had a little bit of this creatine phosphate present all the time. You've also started out in your muscle fiber with ATP, right? Before you're doing stuff, you need ATP for all kinds of processes. You know, the sodium potassium pump needs ATP to run. Any form of primary active transport needs ATP. So we have a little bit in your cells just present at kind of baseline. That happens. If you start to contract that muscle and you start to use that muscle heavily, is you're immediately going to be dropping down those baseline available ATP molecules. Okay. When you were starting out at baseline, you were at an equilibrium, right? So the equilibrium we're talking about here, right, was on one side of our equation. We had creatine phosphate and our ADP, our adenosine diphosphate molecules. And on the other side, we have some creatine and some ATP, our baseline already made ATP that we've been generating kind of at a slower rate than we're about to talk about. Okay. So, in principle, chemistry in the world, something called the law of mass action. So we've talked about this a little bit kind of sideways before, but we haven't really harped on it. What this really says, this law of mass action, is just that like when we have a reversible reaction, right? If you have more of something on one side and less of something on the other side, that's the direction on net that you're going to go, right? So what we do when we are beginning to work our muscles is we are dropping down the amount of ATP we have available, right? So this means that we are now creating the situation where we have more of the things on the left-hand side than we have of these things on the right-hand side. So this starts to drive the reaction in this direction, which is why on the whole, we start to use this creatine phosphate to phosphorylate the ADP. So we've got an enzyme regulating that. That's our creatine kinase. And when we do that, when we take that phosphate and move it over to our ADP molecule, that gives us some more ATP and leaves us with creatine left over here. Okay. So because we have this present, this can immediately give us about a multiple of 5 times the amount of ATP that we had present in the cell at rest. Okay, so this is what we think of as kind of on-tap energy. We didn't require a lot of cellular machinery to do this. We just have that one enzyme in there. This is pretty easy and pretty fast. Okay. So when we think about working out, when we think about exercise, that creatine phosphate is going to be one of our first forms of energy that gets used by your muscle fiber for its ATP production, right? So what we're looking at here is over time, so over minutes of exercise, right? We're looking at how are we making the ATP, right? So this is our energy used per source, right? So which method of making ATP is in charge of this point, is most useful, is being used the most, is giving you the most ATP. Okay. So when we start exercising, the creatine phosphate is our first immediate source of making new ATP molecules. So we've got a little bit of it on top. It gives us 5 times more, it's a fair bit more ATP than we had just hanging out in the cell ready at baseline, but we use it up pretty quick, right? So you can see this steep drop off, right? In this diagram, we got like a minute from that creatine phosphate here. Okay. What we're looking at in this diagram specifically is what type of energy generation you'd be doing if you're doing like light to moderate intensity activity. So as we think about muscle fibers and as we think about energy, we're going to be distinguishing, are you doing something not that strenuous initially that you might be doing for a longer amount of time versus are you doing heavy intensity exercise that you're going to do in bursts, right, and exhaust yourself quickly. Okay. So when we're doing light to moderate intensity activity, and our goal may be doing this for a longer amount of time rather than short bursts, we start out by using that creatine phosphate. And the next thing that takes over is our anaerobic glycolysis, right? So this is pretty purely a product of if you're doing the steps of aerobic metabolism, that glycolysis happens first, and it's in the cytoplasm already. We don't have to go into a mitochondria. We don't have to build up a hydrogen ion gradient. We don't have to do all these things. So this is a little bit faster to get working too. Okay, so our second thing that we do is we start doing glycolysis, our first step of our cellular respiration. And we can do some of that anaerobically, meaning we don't require oxygen. We saw that there were ways we can take that pyruvate and instead of pulling it into the mitochondria, we can leave it there in the cytosol and turn it into lactate so that we regenerate our electron carriers in their oxidized form so that we can then reduce them and keep cycling through glycolysis. So we do a little bit of that in our light exercise, but ultimately what takes over is oxidative phosphorylation. Right. So if we're doing light exercise for a period of time, ultimately we are going to settle on doing our most efficient form of energy generation. We get a lot more ATP from oxidative phosphorylation. We do need oxygen. We do need all this machinery in the mitochondria to be working properly. Takes a while for this to take over. But once it does, super useful, we're going to stick with that. So if we kept going for half an hour, you just see this line kind of extending, right? Okay. So this is the primary method of energy generation in light to moderate intensity activity over time. We do use the other two pieces to kind of kickstart us. If we're thinking about where the glucose for this is coming from, we're going to start out with any glucose that was in the cell to begin with, because you're making a little ATP, but any glycogen, that storage molecule, that polysaccharide in the cell, you're going to break it down into its glucose monomers, and you're going to use them. While you're doing that, your body is going to be pumping your blood around, and it's going to be trying to get you glucose and fatty acids from other places as well. In order to do this, right, in order to end up settling on our oxidative phosphorylation requires oxygen, right? Because it requires oxygen, if you're going to do this type of energy generation, you're going to need to keep those O2 supplies up, which means you're going to need to breathe enough to keep up with how much oxygen you're using. So if you're starting out, you're going on a job, right? You're going to breathe a little faster. You're increasing your ventilation to get more air in your lungs, therefore more oxygen in your blood. You're going to increase your heart rate because that heart is pumping oxygenated blood around your body. So we need to get the oxygen into the blood. We need to get the blood to your muscles. As you're increasing heart rate, we're also going to squeeze faster, right? So not only are we're going to squeeze harder. So we're squeezing the heart faster as well as harder. So more blood is coming out of the heart for every heartbeat. Okay. And we're even going to change some of the blood vessels going to our muscle. So every muscle has blood going to it, blood coming from it, arteries going to the muscle, things coming back. So we're going to dilate those vessels. We're going to make them wider. We're going to increase their diameter so that we can get more blood to a muscle. Once we have the more blood, we've got more oxygen, we can do more oxidative phosphorylation. This is a little different than what happens if you're doing really heavy intensity activity. So things like weightlifting, or if you're still thinking running, this would be like a sprint, right? As fast as you can. So we can see some similarities, right? So we're still starting with our creatine phosphate as our first method of energy generation, right? But what we can see here is that our anaerobic glycolysis is really the big method of energy generation. We do a little oxidative phosphorylation, right? So there's some of it happening, but it's not overtaking glycolysis like it does if you're doing lighter activity. It's really relying on glycolysis primarily the whole time. Okay, so this is a less efficient process, right? You get less energy per glucose molecule if you're just running glycolysis over and over again, but you get it faster. Okay, and we'll see that there are some reasons that we end up relying on glycolysis in muscles that are adapted for this heavy intensity activity. So if we're doing this anaerobic glycolysis, we're starting into our metabolic processes, but as soon as we make that pyruvate, instead of moving it into the mitochondria, we're mostly just turning it into lactate, cycling over and over and over through that first step, through that glycolysis. You're only getting 2 ATP molecules per glucose molecule here. Okay. The reason for this is going to link up to different types of muscle fibers that are used for different types of activity. So we can classify skeletal muscle in a number of different ways. Different muscles are used for different tasks. We know they have different names. Depending on their different tasks, they're going to have different metabolic demands, right? So some of your muscles have to be really strong, have to lift a bunch of weight. Some muscles have to do stuff quickly. Some muscles are doing really, really fast little delicate movements. We're going to have different types of muscle fibers associated with those different tasks. Okay. So when we're looking at those motor units, we just had kind of generalized little stringy, circular, and cross-section muscle fibers. But they're actually different from one to the other, depending on the muscle and depending on the task. So ultimately, we're going to end up with 3 types of muscle fibers. And to get those 3 types, we're going to have 2 ways that we can do contraction, so 2 speeds of contraction. And we're going to have 2 ways that we primarily generate ATP, and we're going to recombine them. Theoretically, if we recombine those, we should get 4, but one of the types is like, don't make sense. I'll show you what I mean by that in a second. But we're going to start out by thinking about contraction velocity, fast and slow. And then we're going to think about ATP production. And then we're going to tie the 2 ideas together. Okay. So thinking about fast versus slow. We're thinking really about how fast can you do the crossroads cycle, right? So a faster muscle contraction, you're going to need to make that sarcomere shorter faster. The way you do that is faster cross-bridge cycling. So this is going to rely on this ATP hydrolysis step. So when I said that you can see it, quote unquote, at the top of our cross-bridge cycle, I had this picture in my head, right? So what we saw in the cross-bridge cycle is that the way we're putting the myosin into its high energy form is we bound on an ATP molecule to that ATP ACE site that caused our myosin and our actin to pull apart. Once they're pulled apart, we can break that ATP molecule into pieces. That gives us our phosphate ion and our ADP here. This pulls in this little cross bridge, pulls in this myosin head, right? Setting us up to restart our cross bridge cycle, grab onto the actin and pull it. So when we talk about fast versus slow muscle fiber types, we're really thinking about how fast can they do this step. And this is going to be kind of hard-coded into a muscle fiber. It's going to depend on like how effective the enzymatic action of that particular myosin molecule is. Now, how short we can make that step, you make it shorter, you can do this more times per minute, basically. So we're going to have fast twitch fibers and we're going to have slow twitch fibers. And they depend on that rate of ATP breakdown. So they depend on the rate of myosin ATP-based activity. Okay. So in a fast twitch fiber, here's what it does on the tip, right? It can cross-bridge cycle faster, which means it can contract faster, and it's going to relax faster. Okay. A slow twitch fiber is the opposite. So that slower contraction is because we have a slower form of myosin molecule, basically. So that enzymatic function on the myosin molecule is a little bit slower. So this is going to be a slower contraction and a slower relaxation. So if we look at named muscles, we can see that in a named muscle, we must have fibers that are faster and slower, because while we always start with a latent period at the beginning of any twitch of any muscle, if you look at an eye muscle, so an extraocular muscle is a muscle that moves your eye, and your eyes are moving back and forth very rapidly if you're trying to like track something, right? You need to be able to follow it quickly. You need to be able to stitch together images quickly. So those extraocular muscles, they twitch really fast, right? So they can contract fast, they can move fast, and they relax fast so that you can do it again, right? So we can see that happening way before something like your gastrocnemius muscle, or in a more extreme condition, even your soleus muscle. So those are 2 of your plantar flexors, right? So 2 of your calf muscles, right? So your calf muscles, you're not going to be able to move, but they don't have to move as fast as your eyes have to move to follow something running across your field of vision. And in fact, you maybe want to keep them contracted for a little bit longer, right? They can help you balance a little bit, right? You're standing on your legs, right? So these muscles react a little slower than an eye muscle would. And especially in the case of the soleus, right? They take a lot longer to relax. Okay? So linking this up to fiber types, you'd expect there to be more fast twitch fibers in our extraocular muscle, right? And a lot of slow twitch fibers in that soleus muscle. Okay? All right. So I thought in mind, our next topic that we're going to link in with this are those methods of ATP generation in different muscle fiber types. So we're going to see that we have glycolytic fibers. They mainly do glycolysis. We're going to have oxidative fibers. They mainly do oxidative phosphorylation. I want you to guess or predict which of these is going to get tired faster. Where are we going to have faster fatigue in a muscle fiber? If we're relying on glycolysis, if we're relying on oxidative phosphorylation. Okay, take a look. Cool. Yeah. So glycolysis is less efficient, right? It produces less ATP per molecule of glucose. So if you're doing glycolysis as your main method of energy production, you're going to get tired faster. That muscle fiber is going to get tired faster than if we're producing on the order of like 30 ATP per molecule of glucose, like we would during oxidative phosphorylation. Now, before we go into that next little bit, one more question for you. So go ahead and look back to those graphs about what we're using for energy in that low to moderate intensity activity versus high intensity. I want you to predict for me, when do you think we're mainly going to be using a glycolytic fiber? What type of activity? Okay, yeah. So the heavy intensity activity was the one that we used a little bit of that threatening phosphate first, and then start doing glycolysis, and we really rely on glycolysis mostly for heavy intensity activity. We did the oxidative phosphorylation mainly with that light to moderate. So we are going to be using these glycolytic fibers, these glycolysis, mainly for heavy intensity activity. And this is all going to create this link between type of metabolism, so the glycolysis that doesn't use oxygen, and these strong forces in large muscle fibers versus smaller muscle fibers that are better for light to moderate intensity activity, but longer term activity, with our oxidative phosphorylation ultimately taking over as our main way of generating energy. We'll take a look at this. But this is at least how I try to remember the trace of a glycolytic fiber versus the trace of an oxidative fiber, and back myself out in my mind to think about what are each of these things doing, and how does that make sense in the big picture of how your body works. Otherwise, it didn't have this right. Okay. So if we're thinking about a glycolytic fiber, okay, so glycolysis only gets us to ATP on net. happens fast because it's out in the cytoplasm, right? But it's not super efficient for energy breakdown. They predicted for me that that's going to be quick to fatigue if you're doing glycolysis, right? So we got that already, all right? We already got that the name already tells us we're doing glycolysis as our energy production. So we're going to link some of these other things to the fact that glycolysis is anaerobic, right? So anaerobic equals no O2, right? No oxygen required here, okay? Which is what links some of these other things together. Not all of them, but some of them. Okay. So if you're relying on glycolysis happening out in the cytoplasm, you don't need mitochondria. So a glycolytic fiber is going to have fewer mitochondria than an oxidative fiber would, because if you're doing glycolysis, this is not particularly necessary. What you are going to need is lots of enzymes for glycolysis. So we haven't talked about specific glycolytic enzymes, and we're not going to. But glycolysis does have several steps within it, I think like 10. They're each mediated by an enzyme, right? So enzymes are catalysts or reactions, there are lots of steps. We need enzymes to do glycolysis. So glycolytic fibers can have them. And jump down to this large diameter part first. Okay. So the large diameter, when you think of it as associated with this strength idea, right? So this heavy intensity exercise, where you're trying to do something hard, but you're going to get tired fast, right? You're getting tired fast in part because you're relying on glycolysis, not super efficient for energy production. Which means in order to do a strong activity quickly, right, it's going to be helpful to have a big, wide, large diameter fiber, because then you can stack those sarcomeres, right, make those stronger movements, more tension, right? You're doing it pretty quick. There's going to be a trade-off there. So a large diameter, if we try to relate that to this oxygen mitochondria stuff, right? This metabolism stuff. Whenever we're thinking about a cell, right? In order to get gases in, gases are nonpolar, or at least oxygen is nonpolar, right? So this is happening through passive transport. So just simple diffusion across our phospholipid bilayer, across our membrane, okay, to get oxygen into a cell, right? So if you have a little cell, you've got a lot of surface area for that oxygen to dissolve across to get into the center of your cell, where our sarcomeres are going to be. But if you imagine a bigger cell, this is great because we could stack lots of sarcomeres, right? So we can make this fiber stronger, but relatively, we've got less surface area for those oxygen molecules to come in. And if we wanted to get those oxygen molecules to the center of our cell, we'd have to go farther, right? So we'd have to travel farther, and the inside of this muscle fiber is less exposed almost to the bloodstream, right? Less of it is in contact with that oxygen. So we've got a trade-off here. Large diameter, nice and strong, lots of force, lots of tension. But a large diameter also makes it difficult to get oxygen, right? So this is why, in part, we have a little bit of a linkage between this reliance on glycolysis, which doesn't require oxygen, right? No oxygen required for glycolysis, right? So if you have a big, strong fiber, lots of sarcomeres in parallel, if they right next to each other, how can you glycolysis if you can't get oxygen all the way into the middle? So this is partially why we have this link between strong movements, lots of diameter, lots of sarcomeres, but this less efficient form of metabolism. The other thing that happens to be true about glycolytic fibers is that since we're trying to do these strong movements quick, we're going to have lots of glycogen in these fibers ready to give us glucose, which is helpful because this is not a terribly efficient process. We're not getting a lot of ATP per glucose molecule, so it's helpful if we have a lot of glucose stored in glycogen. For our oxidative fibers, basically everything's going to be the opposite, right? So if you're relying on oxidative phosphorylation for energy, right? So that's that form of producing ATP that gives you more bang for your buck, right? So we're using that hydrogen ion gradient flooding across to get us lots of ATP, okay? We need oxygen for this, right? So oxidative phosphorylation requires oxygen. So this is going to drive some of these other features that we see. Okay, so in an oxidative fiber, you're going to need lots of mitochondria because oxidative phosphorylation happens in the mitochondria. You're actually going to store oxygen in those fibers, right? So we're doing oxidative phosphorylation inside mitochondria. We have a molecule that can store oxygen called myoglobin. So myoglobin is colored. So myoglobin itself is red in color. I was going to tell you about your turkeys, is that when you're looking at white meat versus dark meat, one of the things you're looking at is whether that myoglobin is present or not. So whether we have that coloration, that oxygen storage for oxidative muscle fibers that can do less intense activities, but for long periods of time. Then we're going to need lots of blood flow to get additional oxygen. So we're going to have lots of capillaries. We want that surface to be in contact with the oxygen. So we're going to have smaller fibers of smaller diameter. And we're doing this for a longer period of time, although the intensity of the activity is lower. So we're going to be resistant to fatigue because we're going to keep feeding these cells oxygen. We're going to keep doing this efficient form of ATP production of oxidative phosphorylation, so we can keep going. So we'll link together these types of fibers from an ATP perspective with our fast and slow twitches.